So I'm going to be talking about the Canadian data that we've collected so far. And I just want you guys to keep in mind that what I'm going to be showing you today is pretty much very similar to what we'll be doing on the smolt samples that many of you have so kindly collected and sent to us. So yeah, keep that in mind as we go through. And I'd be really interested to hear everyone's thoughts and opinions on the metrics that we use at the end of this to look at the vulnerability of different populations to climate change based on their run timing genotypes. So yeah, I'll crack on. So migration is a long-term evolutionary adaptation to seasonally timed events, and it's evolved through predictable environmental cues, whereby individuals migrate from their known local environmental conditions to unknown destination environmental conditions. So they use proxies like temperature and photo period, so day length, as an indicator when to leave their rivers, if we're talking about fish, or when to leave their current environments to then migrate to their destination. But with rapid environmental change, the decoupling is occurring between known and destination conditions. So this might result in individuals arriving at their destination out with those optimal conditions, resulting in population declines. And this is what we're seeing in Atlantic salmon. Populations are declining across their entire range, and they have also a migratory species that's very variable within and between populations. So for example, for their both migratory life history stages, smolts and returning adults, they migrate at different times both within and between populations, and a specific time window as well in which they need to migrate. So for smolts, they've actually advanced the migration timing with climate change, but we don't yet understand whether this migration is phenotypically plastic, so whether they're able to adjust their phenotype without any changes to the genetic basis, or whether it is genetically based. And the same thing with returning adults as well. So they're also advancing their returning migration timing with climate change. So, but we do know that there is a genetic basis to this, with transplant experiments in the past showing that an individual taken from one river with a particular migration timing, moving to a different river with a different timing, it retains its original migration timing of the original river. So there is some kind of genetic basis to this. And if we look at studies on Pacific salmon who look at the genetic basis of run timing, they found the GREB1L and ROC1 genes on a single genomic region to be associated with premature migration. And this has arisen due to a single evolutionary mutation. So if it's lost from the population, then it might not be able to re-evolve again in conservation timescales. And if that population has lost this particular allele that codes for premature migration, then maybe they're less able to respond to climate change and adjust their migration to occur earlier. So this is something really to consider when we're thinking about conservation, but there is also a lot of debate surrounding usage of large effect loci for conservation as well. So this brings us to ask what we know about the genetic basis of run timing in Atlantic salmon. So two studies, one by Eve and one by Vicky, have found a single region with a gene 6.6 associated with run timing. But other than this, like a single study in Scotland and a single study in Finland, like there's not many studies conducted on run timing of Atlantic salmon when you compare it to the Pacific salmon. And this is where SalMove comes in. So SalMove aims to understand how Atlantic salmon populations will be globally impacted by climate change through their migration phenotypes and genotypes. So we've got this amazing array of collaborators across the range of Atlantic salmon. But for today, I'm just going to focus on our Canadian results, where we've characterized the genomic architecture of run timing, and then we went on to examine the genomic vulnerability of those populations to predicted climate change. So these are where our populations are distributed. So we've got 7 populations. We've got the 2 in Labrador, 3 in Newfoundland, and then a couple in the Maritimes area. So a total of 447 individuals, and we use the individual day of return as the phenotype as well. And just to show you what the run timing patterns looks like. So this run timing pattern is based on a 28-year data set, and you've got the mean daily cumulative count on the y-axis and the day of adult return on the x-axis. And each colour corresponds to, so the green is the 1C winter salmon, and then the red is the multi-C winter salmon. So you can see they have very similar distributions in their run timing peaks across those populations. And I don't know if you can really see, but there's a green line underneath, and this is where our genome samples were taken. So you can see they're mainly taken during the peak. I don't know if you can see my mouse, but yeah, most of them are taken within the peak of the run timing. But if we look at the Maritimes area, which are these two here, they have this bimodal run timing distribution, and this is where our samples are biased towards the latter half of the peak. So it's interesting that this area only has this bimodal run timing pattern, but for our association analysis, we include age at maturity and sex as a covariate to try and account for those differences that occur due to differences in sex and age at maturity. So then we go on to our methodology on looking at how we looked at these associations. So we use low coverage whole genome sequencing. So what this is we use a reference genome. So an older reference genome was called Brian. So we'll use Brian's reference genome, and then studies then compare their samples to the reference genome. So it's a standardized look at your genetic variation across individuals, all against Brian. So here you can see that it's low coverage because there's only a certain amount of reads that have been sampled for each area of the genome. And then from this, we get our genetic variation, including single nucleotide polymorphisms, otherwise known as SNPs. So this is a single base pair change in the DNA sequence, which can vary between individuals. And that's what we'll be using as our for our association analysis. So then we correlate these SNPs, our genotype, with the individual day of adult return, whilst accounting for things like sex and age at maturity as covariates. But we also need to consider the fact that Atlantic salmon have very strong natal homing. So this results in really high differentiated populations or highly structured populations. So to try and account for this, we also include population structure as a covariate as well. So to do this, we look at the principal components analysis. So here on PC1, you can see, and PC2, you can see that the Maritimes region is clustered together, the Newfoundland region is clustered together, and then the Labrador region is clustered together. So it kind of pulls out those populations in space, and then we take these axes, PCs 1 to 3, I don't show 3 here, but we take those axes and we use that to correct for population structure. So the results that we're finding aren't merely associated to differences between populations, we then get better insight on what they're actually associated with, so our run timing phenotype. So what we get then is these Manhattan plots. So a Manhattan plot is simply, if you look at Manhattan in New York, you can see these big skyscrapers. And these big skyscrapers are showing us that they're strongly associated with run timing. So on our results, you can see p-value or q-value, because we've corrected for multiple tests on the y-axis, and chromosomes on the x-axis. And on chromosome 17, we had this huge peak, which shows that there's something here that's really strongly associated with run timing. And if we zoom in, you can see that this gene PPFI2 is the highest, it's the most outlier, it's a SNP overlapping PPFI2 that's the highest p-value. And if we look in the literature, we can see that this same gene, PPFI2, has been found associated with migration timing in a long-distance migratory songbirds, as well as in sleep and wakefulness in other birds as well. So really interesting gene, but how can we use this to inform conservation planning? So if we look at how PPFI2, the allele frequency, is distributed across the environment, you can see that we get some kind of clinal patterns as we move northwards. So in the Maritimes, we get a lot of alleles for PPFI2, but as we move northwards, this allele frequency disappears. So we can actually take advantage of this allele frequency decline with the environment and to try and predict how our genes that we found to be important for local adaptation, how they will change with the future environment. So, and without not just simply using one gene, but we'll use all those SNPs that we found to be significantly associated with run timing. And we'll use a database which pulls out 19 bioclimatic variables that are known to be important for biodiversity. So we'll take this data and we'll plug it into a machine learning algorithm called a gradient forest. So we've got 33 SNPs and 7 uncorrelated bioclim variables. And what this does, it builds multiple forests, and at each bifurcation, it takes a different environmental variable and looks at how the allele frequency changes. So you've got these multiple forests, and then they build these decision trees and come up with their own independent tree prediction of the best environmental variables that explain your phenotype, so run timing. And then from that, you get a final prediction, and that's your most important variable to explain run timing. So for us and for run timing, we find the mean temperature of the wettest quarter to be the most important for genes associated with run timing, or the SNPs that we find associated with run timing. And the least important were actually precipitation events. So temperature was a major driver driving this genetic variation. So now we need to use this gradient forest model to also try and predict the future. Like, how are those important variables, like how is the genetic variation going to change with the future environment? So to do this, we use that gradient forest model again. We've trained the gradient forest model in the present data and the present allele frequencies, and then we use the future climate data into that same model to predict our genomic offset estimates. So that's just the difference between the future climate and expected allele frequencies and the present climate. So then we look at how much our allele frequencies need to change to be able to cope with future climate change. And this is what a genomic offset estimate is. So our results from this, you can see that English River has the highest genomic offset. So it needs to change, the genetic variation needs to change the most to match the environmental conditions for the future. So I chose to use the most extreme climate variable, which is in the year 2070 under the worst economic scenario. So that's if nothing happens to our carbon emissions. And so what we get is the most northerly distributed population being the most vulnerable. So you can also see it actually has a late, well, you can't see, but it is also one of the latest is a population with the most latest run timing as well. But we also need to consider other factors. So we've got this great estimate, that's nice, but what about the ability of populations to move? So like, even though these southerly populations, they don't seem to have a high genomic offset, they might be less able to move as the climate changes. So there's more competition for these populations in the south, whereas this population up north is more towards the limit of the distribution of Atlantic salmon. So maybe they have a higher genomic offset, but are more able to move northwards. So we need to consider that as well when using these kind of metrics. And this metric also assumes that populations are currently optimally adapted to their environments, and that might not actually be the case. And we also are lacking a lot of experimental validation for this, especially in animals. So it's just logistic constraints with animals. It's a lot harder to do than in plants. But people have done demographic correlates. So this example can be seen in birds, where she correlated how genomic vulnerability changes. And what they found was genomic vulnerability correlated with population declines. So there is some statistical questions within there as well, but otherwise this is the kind of data that we can use to really inform what our genomic offsets mean and how they translate into real life. Another way we can validate it is the use of old samples. So finklets and scales that I know a lot of you have thousands of, and we can use these to maybe, if we can get a decent genomic coverage, to determine whether what was predicted for those old samples, whether they actually match what's happening today. So we can use that to predict the present and to see how well it managed to predict the present. And then from that, we can have a better understanding on whether our findings are true and how they can translate into real life. And similarly, we can see whether those populations that we predicted to decline, did they actually decline or did they extirpate? So to summarize, we found the strongest association with run timing was for SNP located on the PPFI2 gene, and it's also associated with long distance migration and sleep and wakefulness in birds. The most important variable for run timing associations for the genetic variation associated with it was mean temperature of the wettest quarter. And we found English River to have the highest genomic offsets. So this is the most northerly population, as well as having the latest run timing. So our next step is to expand the current range in Canada but using population level data. So I'm sure many of you have run timing data that goes back years, but no genetic data to correspond to it. So that's exactly what the Canadian government have as well. So what we're trying to do is take that average of that 28-year data set and population level runtiming data and population level genotype data, and use that to try and see if we can associate any kind of to determine whether we get similar associations using a 220K SNP array. So this has a different coverage, and we may miss the PPFIA2 gene, but with conservation money towards conservation management being difficult, like maybe this is all we can afford in some situations. So it's good to understand where we are with the whole genome sequencing data, as well as a smaller array like a 220K SNP chip. And then we'll also use the small samples that many of you have sent to us. Thanks again. And we'll do that for the range of Atlantic salmon. So Atlantic salmon in Scotland, Finland, Norway, Europe, the rest of Europe, and also Canada as well. And for this, we'll use a 60K SNP array. So a lot smaller, but enables us to get a broad look at the associations across the entire range. So yeah, I just want to acknowledge everyone who's helped with this project, providing data, and thanks to the DFO team and the UHI team, and also thanks to you guys for listening as well.